Welcome to the University of Puerto Rico and the Department of Biology here at the University of Puerto Rico's Invertebrate Zoology Laboratory. Here we work with the meiofauna associated to the Caribbean mesophotic reefs and these ecosystems, which have been a new outburst in the scientific community. Over here we have some of the students that work with us. We have Liane Lebron, Isabel Obando, Monica Garcia from Ecuador, Yamilet Vega, Nemesis Flores, Dianette Morales, Zuleika Rodriguez, and Alexander Valle, who is recording this video right now. I'm Juan Carlos Rivera. I have been working in this laboratory for 2 years now. It has been a great experience. I've learned a lot about zoology and more applications towards zoology. Our laboratory participated in the undergraduate research symposium held here on campus last May 2013, and we had the honor and the privilege of winning first place in our category of animal ecology. So let's step into the processes and the studies we have here. So first of all, we have the centrifugation process. Here we have Zuleika. She's pouring 2 milliliters of sediment that has been brought up from the mesophotic coral. She pours 2 milliliters of sediment and 10 to 12 milliliters of Ludox AM, which is a colloidal silica suspension. After she does this, she places the 6 centrifuge tubes into the centrifuge itself and allows it to go for 2.5 minutes, 3,500 revolutions per minute. Thank you, Zuleika. Here we have our students sorting the organisms. These organisms, mind you, are the supernatant that were obtained from the centrifuge tubes. This makes it easier for them to find the organisms and classify them by taxa and organize them, then count them. Here we have Yamilet Vega working with some ostracods and a few other organisms.
I found an ostracod.
She found an ostracod. The ostracods are the most special organisms that we can find here because our graduate student, who is our boss, Alexandra Galindo, she's doing her master's thesis on these species, which very little is known about them. So come on in and check it out. So, after they're done sorting and classifying the organisms, they come over to the sink and they sieve the rest of the sediment that they worked with. Here we have Dianette working with the system. All the sediment that was left over, there are no more organisms found in that sediment. She will just sieve it through a 0.155 millimeter sieve, as you can see. And she is using 95% alcohol. All this goes back to another centrifuge tube, which will later on be reanalyzed to make sure no other organisms are left over. And finally, the last process in all of this is counting the organisms. Once these organisms have been classified and placed into different tubes, we count them in their different groups, taxa. This is why we have Isabel Obando. She is counting a few organisms. As you can see, she has a few tallies. Come on closer. So now we'll be interviewing Janet Morales, one of our fellow students who has been working with us since last semester. She will tell us a little bit more about her experience here at the lab. So tell us what you are majoring in, your year of study, and a little bit more about yourself and your work here.
Pues mi bachillerato es en microbiología industrial, el primero, y tengo un segundo bachillerato en biología. Terminé ahora en mayo, ya que estoy en mi 5º año, y llegué aquí en un día de ajuste. Estaba buscando una investigación y me topé con la profesora Mónica Alfaro. Ella me habló sobre la investigación, sobre lo que estaban haciendo, y me interesó mucho, ya que siempre me ha gustado mucho lo que es la ecología y el mar, así que era algo nice, algo bueno y innovador que hacer.
So, what have you learned while you've been here in the lab? Have you learned more about ecology, zoology, or some type of topic in depth?
Sí, he aprendido un poco más sobre la zoología, sobre los invertebrados marinos, ostracos, caneláceos, isópodos, y también he podido aprender un poquito más sobre cómo manejar estos microscopios, así que he aprendido un poquito de todo.
Okay, that's great. So, what are your plans for future after graduation?
Es un poco curioso, ya que no voy a seguir con la ecología marina, voy a seguir con lo que es medicina, pero como siempre la ecología me ha gustado, así que quizás en el futuro me vea ayudando un poquito en esta área, ya que se han encontrado que sustratos de corales y algas se ven con importancia médica, así que quizás en el futuro investigue un poquito más en esa área.
That's great. So, thank you very much. Thank you for your time. You're awesome. And that's it for now. And now we want to see a different perspective on how working in this lab has gained, how our students have gained some knowledge from working here. So we have Monica Garcia, who's from Ecuador. Tell us, what are you majoring in? What's your year of study? And what knowledge have you gained from working?
Well, my major is biology, and I'm currently in my 3rd year here at UPRM, and knowledge, basically I wasn't familiar with any of the organisms we're studying, and it's eye-opening. It's really amazing everything we do in this lab.
So, you've learned a lot. You've learned about the organisms you never thought existed. So, what are your future goals after graduation?
Well, thanks to this lab, I realized that marine sciences is something I'm very interested in, so I'm planning on applying for a PhD, and I will continue here at UPRM, and those are my future goals.
Awesome. Thank you very much for your time.